In this video, we will be discussing about the process of DNA replication. DNA is a molecule made up of two strands twisted around each other in a double helix shaped. Each strand is made up of four chemical bases represented by letter A, C, G and T. These two strands are complementary. Whenever there is T in one strand, there will be A in other strand, and whenever there will be C, there will be G in other strand. These two strands are complementary and anti-parallel to each other. Starts with 5' end to 3' end. The first step in DNA replication is to separate the two strands, and an enzyme called DNA helicase, also called as unwinding enzyme, does this process. This results in formation of replication fork. This creates two separate templates of DNA. Now, an enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This is the starting point of the construction of new strands of DNA. Now, an enzyme called DNA polymerase binds to primer and will make new strand of DNA. This DNA polymerase can only add bases in one direction, that is from 5' end to 3' end. Now, the leading strand is made continuously, whereas the DNA polymerase adding bases one by one in 5' to 3' direction. The other strand, which is called as a lagging strand, cannot be made in a continuous fashion because it runs in opposite direction. The DNA polymerase therefore binds only one time, make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with RNA primer. DNA polymerase then adds a short row of DNA bases in 5' end to 3' end direction. The next primer is then added further down the lagging strand. Now another Okazaki fragment is then made and the process is repeated again and again. Once the new DNA has been made, the enzyme exonucleases remove all the DNA primers from both the strands of DNA. Another DNA polymerase enzyme then fill in the gap that are left behind with DNA. Now, finally, the enzyme DNA ligase seals up the fragment of DNA in both the strands. This is how DNA replication is described as semiconservative because each DNA molecule is made up of one old and one new strand of DNA. This was given by Michelson-Stahl experiment. So thanks for watching. Do like, share and subscribe.